So now moving on, looking at things when things have progressed a little bit more, right? So when we look at this one, this is just when things are getting started, but there's more to it. And so we're looking at kind of a combination between the last two, right? We've looked at this one before, but now we're zooming into this section right here, and we're looking at the proteins that are involved in this portion of the replication. And so we have two new proteins that were not on the previous one, and that's another type of DNA polymerase. And DNA polymerase 1, so remember DNA polymerase 3 adds the new nucleotides in. DNA polymerase 1 is going to function by removing all of the RNA primers, because in the end you don't want RNA in your DNA, so you have to get rid of those RNA primers. And DNA polymerase is the one that is going to do that. DNA ligase, once you've gotten rid of all the RNA primers, you still have a little missing phosphodiester bond between all the little fragments. So DNA ligase is going to come in and join all of those little fragments. So to ligate something is to kind of tie it together. So it's going to join those smaller fragments. So, armed with all of this, you should be able to go through and label all of the proteins, the leading strands, the lagging strands, the parent strands, also not imaged, but you should be able to put it in the right spot, is that topoisomerase. And then where are the 5' and 3' of the daughter strands. So pause and see if you can label all those. All right, so let's start with the thing here that is unwinding the DNA, right? That is the thing that is breaking up the helix. So that is the helicase. And then once it's open, right, you can start having these little bits that are going to get everything started, right? Those are the primers, specifically the RNA primer. The thing that makes that is the primase. And then the thing, so now again over here you have a primer. So this is another primer. But then the thing that's now going to, see now these were red and now these are blue because that's different. These are the DNA that are coming in. The thing that's going to add the nucleotides is the DNA polymerase, and specifically DNA polymerase 3. And what you'll note here is that right there's a DNA polymerase 3 here and a very similar looking protein over here. Those are both the same, because there's more than one functioning at a time. So this is also DNA polymerase 3. Because this strand is synthesizing at the same time that this strand is. And you wouldn't want to have to wait for a new DNA polymerase 3 to come around. So they're happening at the same time. And there's a section in here where for a little while the DNA is single-stranded. So these single-stranded binding proteins are on both of the strands as well. So single-stranded binding proteins are the tic-tacs that are functioning there. The parent strands are the blue ones on the top. The daughter strands are the, well, they're all blue, but the parent strands are the dark blue. The daughter strands are the light blue ones coming in on the inside. The one that is synthesized continuously is the leading strand. Oops, and I forgot to have leading strand in there. So the leading strand is the one that is synthesized continuously. The lagging strand is synthesized in fragments. And let's talk about the 5' and 3' for these. So we've kind of already mentioned you can see how the arrows go, right? And remember, the new nucleotides are always added at which end? The 3' end. So the arrows are always the 3' end. So what you can see if you kind of translate this to this top one is this one here, so that's this one. And so it's being synthesized this way. So this end is the 3' end for this one. And so that means the 5' end is over here. And then this one, right, the primers are all over at this end, and then the arrows are all over towards the right. So the 5' end for this one is here, and then the 3' end is over here, because that's where the direction that it's going. Because all the new nucleotides are added on this end to the right. And then our topoisomerase is the thing that's going to help prevent the strain by breaking and swiveling the DNA. That's going to function ahead of the helicase, so it's going to be where that parent strand is still double-stranded. Not, again, not pictured here. But then now we've got two other proteins, so the two new ones that we've added that we haven't seen before are these two over here. So this one, you can see that here there's RNA primer that we're getting rid of, and so the DNA polymerases are the ones that are bringing in new nucleotides. DNA polymerase 3 is bringing in totally new nucleotides coding off of the parent strand. DNA polymerase 1 is replacing the RNA nucleotides with DNA nucleotides. So this one here is DNA polymerase 1. And then this protein right here, you can see that there is a missing bond right here. And what type of bond is that within one DNA strand? Exactly, the phosphodiester bond. And so this enzyme here is synthesizing that very final bond. It is ligating it. It is tying it together. That last one is DNA ligase. So that is the whole picture for DNA replication. Remember that DNA replication is happening during that S phase when we are duplicating the chromosomes so that those chromosomes can then be split into different cells.